Hello friends, welcome back to my channel Plant Pathology Pathsala. So in this video we will discuss about Phomopsis Blight of Brinjal. This is a very important disease. Definitely you will get one question in any type of competitive exams. Okay, so let's start the video. So as you know that this Phomopsis Blight, it is a very important disease of brinjal, and the pathogen causing this disease is Phomopsis vexan. But as per the Kirkitel, this Phomopsis actually it was under the Deuteromycetes. But as per the new fungal classification, the uh, Deuteromycetes fungus, the imperfect fungus, those who have only the asexual phase, the conidia, are removed actually. They are actually placed either under the Basidiomycetes or in the Ascomycetes fungi based on um, its gene relationship, means its similarity based on rDNA analysis. Okay, sequence analysis, based on sequence similarity, they have placed the Deuteromycetes fungi are either placed under the Basidiomycetes or in the Ascomycetes. So based on the phylogenetic analysis, this Phomopsis was then based on this Kirkitel classification, new classification, the modern taxonomy, it was placed under the Ascomycetes fungi and it was called as Diaporthe vexan. So this is the sexual stage. Okay, so remember this is very important. The pathogen causing Phomopsis blight of brinjal is Phomopsis vexan, and as per the modern taxonomy, its sexual stage is Diaporthe vexan. Then about the key symptoms produced by the Phomopsis blight of brinjal. Here this pathogen actually produce symptoms all over the aerial plant part, in seedling, leaves, stem or petiole, fruits. Okay, so all over the aerial part, diagnostic symptoms you can observe. First, in the seedling, so at the in the seedling, stem lesions near the soil line. Here you can see lesions are formed near the soil line. Here the soil is there, so just at the collar region, brownish lesion will form that later girdle the stem. The stem will become weak and finally the seedling will fall, and ultimately the seedling death will occur. Okay, this is at the seedling stage. Then at the leaf, see, in leaf, circular or irregular grayish brown spots are formed, which later coalesce and they will cause blightening, and ultimately leaf will defoliate or leaf will fall. Then about the stem, so in stem also you will found elongated brown lesions or cankerous things that will later result in drying of the shoots. Okay, here you can see brown lesion or cankers on the stem. And the fruit, in fruit the most characteristic symptoms of this Phomopsis blight were observed. In fruit you will get sunken pale brown lesion. That lesion actually then aggravate and it will ultimately lead to fruit rotting. The fruit will become black and mummified. And most characteristic symptom here on the fruit is the production of black Pycnidia, that in the concentric ring form. Remember this. Here you can see rings of Pycnidia. The black Pycnidia are there. Remember this. The characteristic symptom of blight, Phomopsis blight of brinjal, was seen in the fruit, sunken pale brown lesion that leading to fruit rot and black mummification. That black mummification is nothing but the black Pycnidia, the sporulating structure, the conidia producing structure on the older lesion of the fruits. Otherwise, this Phomopsis blight means infect all the aerial part of the plant starting from the seedling stage. So it will infect the seedling at the soil stem region at the soil line. Later it causes girdling and ultimately seedling death will occur in both stem as well as leaf as well as fruit. Brownish circular or irregular lesion will form in fruit. That lesion develop to fruit rotting and ultimately black mummification, where you will observe the black Pycnidia. Okay, so key identification is sunken fruit lesion, black Pycnidia and mummified fruit. Then about the pathogen, its etiology and the disease cycle. Very important thing. So about the pathogen, it was traditionally known as Phomopsis Vexan. But as I have already told, as per the modern classification, this Deuteromycetes fungi are regrouped either under the Ascomycetes fungi or in the Basidiomycetes fungi based on its phylogenetic relationship. So this modern classification, as per the modern classification, it was renamed as Diaporthe Vexan. It was a Coelomycetes fungus producing Pycnidia. Coelomycetes means dark color. Then Pycnidia. Pycnidia are dark, immersed or erupent in infected tissue producing conidia. Pycnidia is a asexual fruiting body that produce pycniospore or the conidia. Okay, these are generally flask shaped structure. This flask shaped structure where conidia are formed. Okay, so about the conidia. Conidia are highly unicellular, elongated, subcylindrical and responsible for the secondary spread. Here the conidia is there. You can see it is highly unicellular, elongated, subcylindrical and it is responsible for the secondary spread. Then source of inoculum. This is the very important part. Definitely you will get question from this. The primary source of inoculum for Phomopsis blight of Brinjal is infected seed and crop debris, and secondary spread or secondary source is the conidia from the Pycnidia. It is a seed-borne nature. Phomopsis blight, Ascochyta blight, remember these two disease. These are internally seed-borne disease. The pathogen is predominantly internally seed-borne. It is present inside the seed, especially in the seed coat and internal seed tissue. Okay. So let's see this disease cycle. So disease start from infected seed. As I have already told, it is an internally seed-borne disease. So primary inoculum comes from the seed, or it may either present in the plant debris. Then it will infect the seedling. In seedling, I have already told, I have already shown in the photo also that it causes lesions in the stem region that ultimately girdle the stem. Okay. Then it will further infect the leaves. In leaf, stem, fruit, you will get circular to irregular brownish lesions. In fruit, later it will cause fruit rotting and black mummification. In that mummification, you will get this black Pycnidia. Okay, and this Pycnidia is the asexual fruiting body that will produce the enormous number of conidia, the highly unicellular elongated conidia that is responsible for secondary spread. And the secondary spread of this conidia occurs through rain splash or water or through irrigation. Okay, then this conidia will further infect other plants and ultimately it is causing the economic loss. that's why this disease is very important, because it is ultimately affecting the economic product of the plant, the fruit. Okay, so disease in one line, that is infected seed or plant debris causing the primary infection. Then after primary infection, lesions, brownish in the circular irregular lesions will form. Then on fruit, Pycnidia will be formed that will produce enormous number of conidia, which were later spread by rain splash of water and causing the secondary infection, causing the fruit and fruit rot and mummification. That later stages, that from the rotted fruit or mummified fruit, you will get the infected seed, and this disease plant debris, they will later act as the primary source of inoculum. This is about the pathogen, etiology and disease cycle. Then the importance of the disease. So this disease is very important. Why? Because it is affecting the economic crop. It affects leaves, stem, fruits, but fruit infection is most economically serious, because ultimately the farmer, why they grow this plant? Because to sell the fruit only. If fruit become mummified, the rotting is there, nobody will buy that fruit. That's why it is very important. The infected fruit become rotten, black and mummified, leading to loss in marketable yield and quality. The disease can cause 30 to 50% yield reduction under favorable environmental condition, and it is important in both the tropical and subtropical region. Then about the epidemiology, the favorable condition for the development of this disease. So disease development and spread is favored by the high rainfall, frequent rains, because splashing of rain disseminate the conidia. The secondary spread occurs through conidia, which were spread by rain splash and water. Then high humidity is required, because prolonged leaf wetness is required for germination of the conidia. Then warm temperature, about 24 to 28 degrees Celsius and about 26 degrees Celsius is most favorable under wet condition. Then dense canopy and pore aeration, because it creates microclimate that is favorable for the infection. Then the infected crop debris, it helps in survival of the pathogen and build up of inoculum. So these are the favorable conditions. Remember, warm temperature, high humidity, high rainfall. Okay. Next about its Indian status. So it was reported from major brinjal growing regions across India. The seed-borne nature of this disease make introduction through planting material particularly important, and disease severity is strongly influenced by temperature, rainfall, number of rainy days and high humidity. See, almost in because in Indian condition, most of the area are subtropical and tropical regions, so most of the area wherever the brinjal are grown, this disease is reported. Next, about the management. See, before discussing the management, keep the things, I will tell some quick points that you should remember. That first, before deciding the management practices, first think that where the primary source of inoculum comes from. Here for the Phomopsis blight, the primary source of inoculum is the infected seed, because it is internally seed-borne, and the crop debris. So our major focus should be to reduce or to reduce the seed inoculum, or we can just omit the seed inoculum. How we can reduce that infected seed inoculum? By using healthy seeds, or we can also use the resistant variety. So we can use certified disease free seeds, Do not collect seed from diseased plants. This is the major one. Then we can use the resistant variety. Grow recommended resistant and tolerant cultivar wherever it is available. Next, another thing is, if some contamination is there, we can do some seed treatment to reduce the seed inoculum. How? Because it is internally seed-borne, we can do the hot water treatment. So at 50 degrees Celsius for 30 minutes we can do hot water treatment, and apart from this we can also do fungicide treatment to reduce the seed-borne infection. So these three major points we are doing to reduce the seed-borne inoculum. Next, another primary source of inoculum is plant debris. To reduce that, we have to do the field sanitation. Remove and destroy infected plant and fruits. Remove infected crop debris after harvest. Maintain good drainage and aeration. This is for the reducing the inoculum that comes from the crop debris. Okay. Now we have covered everything. So we have checked the primary source of inoculum. Still, if we are getting some infection in the field after checking the primary source of inoculum, means definitely it is coming from some other fields. It is the secondary infection is coming. So how we can manage the secondary infection? For managing that secondary infection, we will do the fungicide application. So need-based fungicide application like mancozeb, carbendazim we can do, and follow recommended dose and latest registered label for the region. Okay, remember this. For most of the foliar fungi, foliar pathogen, we can use the mancozeb and carbendazim. This is common fungicide we can use. Okay. But most important thing is about the seed and management of the crop debris. Whenever in any interview they have asked about the management, first tell about this that this is a internally seed-borne disease. So our main focus should be on management of the seed-borne infection that can be managed through using healthy, disease-free certified seed, or by using resistant cultivar, or we can do some seed treatment like hot water treatment or fungicide treatment. Then for the management of crop debris, we can do the field sanitation. Then some important quick one-line question. The causal organism of Phomopsis blight is Phomopsis vexan. The modern name is Diaporthe vexan. Host is Brinjal. Most economically affected part is the fruit. The characteristic symptom is sunken pale brown lesion that ultimately lead to fruit rot and black mummification with Pycnidia. Then diagnostic sign for this pathogen is black Pycnidia in the older lesions. Then fruiting body is Pycnidium that produce the conidia. Type of seed-borne, it is predominantly internally seed-borne. Very, very important. Repeatedly I am telling this is very important. Okay. Then primary source of inoculum is infected seed and crop debris. This is also very, very important. Then secondary source of inoculum is conidia produced in Pycnidia. Then dispersal of inoculum is through rain splash and water movement. So I think now you are very much clear about the Phomopsis blight of Brinjal. Still, if you have any doubt, let me know in the comment section. So thank you for watching this video.